Welcome to The Explainer. Today, we're diving into something absolutely wild, the biological mechanics of how organisms actually respond to their environments. I mean, have you ever stopped to wonder what happens in that literal split second between experiencing something and your brain deciding, yeah, I'm going to engage with this, or nope, I am shutting down completely? Well, we are going to map out that exact journey today.
Okay, let's take a quick look at our roadmap. We've got 1. The brain's hidden bottlenecks. 2. When sweet becomes good. 3. The 2E cognitive freeze. 4. Tracking the invisible shift. And finally, 5. The internal Lego therapist. We're going to connect some totally fascinating dots here, zooming all the way in on a tiny fruit fly's sweet tooth, and then zooming way out to the incredibly complex cognitive architecture of twice exceptional, or 2E, human minds. Let's get right into it.
Section 1. The brain's hidden bottlenecks, bridging sensory input and behavioral output. So, let's start with the foundational idea. Our brains, they aren't just passive video cameras objectively recording the world. They are constantly and actively interpreting, filtering, and assigning value to literally everything we encounter.
Which brings us to this. How does a brain decide to act, learn, or just completely freeze? This right here is the ultimate question of behavior, because it's not really about what you sense, it's about what that sensation actually means to you in that exact moment. And to figure that out, we have to look at exactly how raw information is routed inside the nervous system. Here is the key. All that information has to pass through these really high-stakes checkpoints. We call them neural bottlenecks. Think of it like this massive funnel, or an hourglass circuit motif. You've got this absolute flood of high-dimensional sensory data, sights, sounds, tastes, your own internal stress levels, all squeezing down into this physiological choke point. It's the ultimate gatekeeper. It takes all that messy data and translates it into a simple, lower-dimensional value signal. That signal is what tells an organism in real time whether to feel rewarded, paralyzed, or fully engaged.
Moving on to Section 2. When sweet becomes good, the fruit fly's reward bottleneck. Let's actually look at a real-world case study to see how this bottleneck operates inside the taste circuits of a fruit fly. To see this in action, we're looking at the Drosophila fruit fly, which, loves sugar just as much as we do. Recent research from Current Biology found exactly where this bottleneck sits. When a fly tastes sugar, that raw sensory data shoots over to a region called the subesophageal zone. And researchers found this specific pair of inner neurons called Fox neurons. These guys act as the literal switch. They form that hourglass bottleneck, taking the raw sensation of sweet and actively assigning a positive, rewarding value to it before sending it off to the brain's learning centers.
Now, look at the distinction here, because it's super important. You have intrinsic valence, which is really just the static, built-in fact that sugar is sweet and generally appealing. But then you have affective valence. That is the dynamic, right now, real-time motivational signal that screams, yes, this is amazing right now, keep eating, remember this. The Fox neurons, they create that affective valence. They don't change what the taste is. They completely change its motivational power. And researchers prove this by looking at 3 core biological criteria. First, sufficiency. If you artificially turn these Fox neurons on, the flies just start eating voraciously. Second, necessity. Turn them off, and while the fly still knows it's tasting sugar, that intense drive to consume it, totally gone. And third, this is the crazy one, override capacity. Activating these neurons can actually override a fly's innate disgust for something bitter, like quinine, making them eagerly eat it anyway. So yeah, these criteria totally prove that Fox neurons are the specific bottleneck transforming raw sensation into a powerful reinforcing signal.
All right, Section 3, the 2E cognitive freeze, the human biological choke point. Let's pivot from the physical bottleneck of a fly's taste circuit to something way more complex, the cognitive bottleneck found in human twice exceptional, or 2E individuals. These are folks who are highly gifted, but also navigate learning differences like ADHD or dyslexia. When a 2E brain is forced to deal with flat, rote, one-dimensional tasks, think sequential text decoding, it doesn't just get bored, it hits a literal biological wall. Here's what actually happens. Bam! An environmental stress spike hits. That triggers a rapid epigenetic change, specifically a chemical modification called M6A RNA methylation, deep in a routing hub of the brain called the supramammillary nucleus, or the SUM. Usually the SUM sends out these nice rhythmic theta waves to organize thoughts, but under this stress, it snaps into survival mode. Those theta waves completely break down. And because of that, Brodmann area 6,
The premotor cortex that plans action and speech gets cut off and literally slams on the brakes. The end result is a full-on physical and cognitive freeze.
And underneath that freeze, it is a brutal chemical cascade. It's like this biological seesaw of stress and motivation. The 1D task stresses the brain out, flooding it with cortisol. That high cortisol aggressively wipes out serotonin, throwing mood way off balance, and it actually numbs the dopamine receptors. So any sense of reward or capability is just stripped away. The brain sees this massive dopamine crash as a severe energy deficit, which forces the adrenal glands to pump out even more cortisol. So, the SUM is actively shut down by this cycle. It creates this heartbreaking disconnect between the 2E individual's brilliant internal capability and their frozen external performance.
Section 4. Tracking the invisible shift, reading somatic signatures with Wi-Fi. So what if we could actually see this invisible bottleneck in real time? Like validating exactly when someone freezes up and exactly when they break free, without strapping them into a bunch of wearable sensors.
Well, using wireless sensing systems, literally using AI-driven Wi-Fi signals passively bouncing off a person's body, we can map this exact shift, and the contrast is absolutely staggering. During that 1D flat line of rote learning, the Wi-Fi picks up compressed, shallow breathing and completely unsynchronized heart rate variability. The SUM is totally dormant and cortisol is spiking. But watch what happens when you introduce highly complex, systems-level logic, what we call 4D arousal. The biometrics totally flip. The Wi-Fi detects deep, regularized breathing, stabilized heart rhythms, and high vagal tone. Suddenly, the SUM wakes up, triggering a huge dopamine spark that clears out all that cortisol.
The crucial takeaway here is that this Wi-Fi tech acts as a real-time validator. In a room where linear thinkers might show perfectly steady biometrics during a rote task, the 2E individual stands out like a beacon. The Wi-Fi algorithms capture their extreme physiological volatility. It validates the exact moment their uniquely wired mind breaks free from those linear constraints and just ignites into a multidimensional flow state.
Bringing us to Section 5, the internal Lego therapist, designing information to fit the brain. If the SUM bottleneck is freezing up because of flat data, how on earth do we fix the data?
That brings us to this incredible quote and principle from researchers at the University of the Witwatersrand on iterated learning. They proved that information must evolve to fit the brain, not the other way around. Basically, complex data has to shed its randomness and take on structure, or compositionality, to get through our learning bottlenecks. For a 2E brain, one-dimensional rote learning, it's just unstructured noise that gets hopelessly jammed in the bottleneck.
So, we can actually think of the SIM as the brain's very own internal Lego therapist. Think about it. A real-world Lego therapist uses physical 3D blocks to help someone communicate when linear words fail them. Biologically, the SUM does exactly the same thing. It aggressively rejects flat, one-dimensional thinking, but throw in highly complex spatial 4D logic, like a torus model of interconnected variables, and the SUM lights up. It translates that spatial manipulation into those synchronized theta rhythms we talked about, letting the 2E brain build abstract concepts using 3D mental models. This engagement kicks off a massive dopamine-driven flow state that instantly slams the brakes on the cortisol panic.
We're literally talking about the real-time evolution of data. By swapping out flat 1D noise for highly structured 4D logic, we aren't fruitlessly trying to force the brain to widen its bottleneck. You can't do that. Instead, we let the environment evolve to match the human brain's natural compositionality. This lets the information bypass the SUM freeze instantly, mapping perfectly onto that individual's totally unique cognitive geometry. Which leaves us with a really profound question. Whether we are looking at a tiny cluster of fox neurons in a fruit fly assigning value to sugar, or the SUBEM in a brilliant human mind hunting for complex spatial logic, our brains are undeniably governed by these hidden bottlenecks. So, if we know that structured, multidimensional information is the ultimate key to unlocking these pathways and stopping the freeze response, how must we reshape our modern learning environments moving forward? How do we stop forcing uniquely wired minds through flat, one-dimensional tunnels and instead build environments that actually unleash their multidimensional flow? Something to really think about. Thank you so much for joining me on this explainer. Keep questioning the structure of the world around you,